I think about a lot of work that we are doing in evolutionary biology, for example, where we just compare genomes and try to understand the extinct species, etc. So then, how much we can rely on that kind of information? I mean, of course, we are getting to know more about the hardware. Then, what about the software? Are there ways to do it or go there?
I mean, you mentioned extinct species, but this is one of the most important things about paleontology. Is the only way you know what a dinosaur looked like is from finding a fossil of it. Most of the variety of life over the last 500-600 million years or so of animals alone. The only way we know what they look like. I mean, in principle, we could do it from the genome in the end, but in practice, we're a long way from being able to do that now. And I don't know if we could ever do it. In principle, if we didn't have examples of the kind of thing to compare it with. So I think we're quite shockingly ignorant about how this system works from a genome level at the moment. And it's I think part of it is we're exactly what Mike was saying. We're forgetting the software. We're looking at the hardware only, and we're not thinking about how is this working as a unified system, as an organism with physiology, and what makes that physiology operate at a higher level to just the hardware that generates it.
Yeah. This business of predicting, you're absolutely right. One of the things that I show in some of my talks is here's a baby axolotl. It's a salamander, and they have little legs. The babies have legs. And here's a tadpole of a frog, Xenopus. They don't have legs. And so in our lab, we make a frogalotl. 50% axolotl, 50% frog. Right. So now you get a frogalotl. So now I ask a simple question. You have the genome of the axolotl fully sequenced. You've got the genome of the frog fully sequenced. Is the frogalotl going to have legs or not? We have absolutely no idea. There's no way to tell. We have no way to tell. And if that's not bad enough, like Nick was saying, if you don't actually have something to compare a genome to, we don't know what the frog or the axolotl would look like either. We have no ability right now to look at a genome and say, yes, this thing has 6 legs and whatever. No, you have no ability to do that other than comparing it to other ones that you've had. So, and not to mention all of the scenarios in which, and we've shown many, and other people have shown many scenarios in which the genetic information literally misleads you, because if there's interventions at the physiology level, that tends to win. And so we have many examples where there's oncogenic mutations, but no cancer. There are developmental mutations, but there's no defect, because we've intervened at the physiological level. Or we have worms that have the heads belonging to another species of flatworm that's 150 million years distant, and you sequence that genome, you will not get the right answer about what the head of that animal looks like. You just won't get the right answer. So, both are important, but it's not all about the genome for sure.
Yeah, so let's kind of talk a little bit more about the software. So is it only about bioelectricity? Maybe first you can introduce a little bit about like what is bioelectricity and how we go about it.
Mike, do you want to go ahead?
Sure. Okay. So where I take up, so to answer the second part is it's I don't think it's only bioelectricity, because there are some interesting, for example, recently we and Richard Watson, some other people before that, found, for example, that very simple things like gene regulatory networks or pathways. It could be a protein pathway. They actually have a kind of plasticity where they can do many different kinds of learning. So they can do habituation, anticipation, associative learning, just in the dynamical systems sense, without any rewiring. So even chemical pathways can do some of this, right? So some of this cognitive stuff goes all the way down. But what I really like about bioelectricity, and where I take it up is sort of after I sort of assume the cell is there and everything is there. So Nick covers the earlier stuff. I kind of just assume it's all there. And after that, what we see is this remarkable thing that because cells have the ability to generate a resting potential using ion channels and pumps in the membrane, and they have these little electrical synapses known as gap junctions, which allow them to share their state with their neighbors. And both the channels and the gap junctions can themselves be voltage sensitive. So now what you have is a voltage gated ion conductance, which is basically a transistor that gives you a kind of historicity. It gives you feedback loops, memory. Right there, right, right there. And when these cells connect with each other into electrical networks, you get some of the same phenomena we see in the brain, just on a slower time scale. So these are not millisecond; these are hours, on the scale of hours. But there's all kinds of amazing information processing that these networks can do that allow them to pursue larger goals. So I love the evolutionary electrochemistry and the metabolics, because I think these are the very first kinds of spaces in which living systems solve problems, right? Metabolic space, bioelectric space, like that. But after that, they can, when they join into these electrical networks, now they can solve problems in anatomical morphospace. They can do things like remember what the correct shape is, figure out, do we have the correct shape, what needs to be done, all of that. And then later on, you get a brain, and now you can operate in three-dimensional behavioral space and linguistic space, who knows what else. So that's my story of electricity, is this like transistor-like ability of cells to join into networks and compute after that.
And can we reduce it to molecular level?
Yes, I think we can, but I don't know that we have the ability to do it yet. I mean, I tend to think in terms of mitochondria and how they're going to communicate. For example, just take a really simple thing. There's two ways of seeing this: apoptosis, controlled cell death. The mitochondria are involved in that, and loss of membrane potential is part of what's happening. Also, whether mitochondria get targeted to be broken down or not. No one's ever quite got what's happening. And it's partly because the mitochondria have two membranes, and the outer membrane is not electrically charged, or at least not very much, if it is at all. Whereas the inner membrane has got a pretty high potential on it. But the proteins that are targeting the mitochondria without a membrane potential are targeting the outer membrane. And there are protein complexes that join the two up, and it's possible that it can work that way. But it's also possible that there's electrical signaling across those distances. And I think it's likely that there is. But then the question becomes, well, if it's simply an electrostatic potential, then it falls off very quickly, because it decays with the square root of the distance. So it's not going to signal very far. Whereas electromagnetic fields potentially could signal much further, but that requires a very specific morphology to be able to work at all. And there's an interesting and difficult question about, well, just what could they do? How can they interact with the hardware? And there are ways that they can, undoubtedly, but is it strong enough? Can we measure it? Do we have the facilities to get at all these things? So I think it does work at the molecular level. And I think over very short distances. So, for example, with the ATP synthase, there's a beautiful work from Wayne Frasch recently showing, I don't know, the C ring, which is it's basically, this is a rotating motor protein that sits in the inner membrane of the mitochondria. And there's in our cells, there's 8 subunits in this ring, and a proton binds on, and it just kind of moves around. So it needs, each proton that binds on, it needs to move. For 8 subunits to turn in a complete circle, that would be about 37 degrees or something like that. So the binding of the proton moves the whole ring around by about 37 degrees. And how does it do that? Nobody has ever quite figured it out, but it's been known for decades that the actual electrical potential on the membrane does quite a lot of that work. And he's shown quite recently that actually it with just the proton, it moves about 11 degrees, and with the electrical potential as well, that takes it another 25 degrees or something of that order of rotation. So it's this local electrostatic charge which is driving that. So over short distances, it's really beautifully demonstrated that you need the charge on the membrane for this to work. But over longer distances, if you're dealing with perhaps a few micrometers or something, then I would be surprised if electrostatic charges alone were doing that. They certainly are at the plasma membrane in terms of connections between cells and having the same charge across effectively wider expanses. But the mitochondria are interesting in that sense as well, in that the cristae are closed very often and separate from each other. And so you can have different membrane potentials in different cristae in the same mitochondria. You can have individual mitochondria, or you can have a network that all join together. There's so much subtlety in all of this, and to underpin all of that, nobody has ever successfully measured the membrane potential of mitochondria accurately. And the reason for that is, if you simply use a microelectrode and you insert it into the matrix of the mitochondria, into the middle of the mitochondria, what you'll measure, and this is work going back decades from a guy called Tedeschi, but other people have come to the same conclusion, you get about 10 or 20 millivolts. It's not much, somewhat less than the plasma membrane. All of the calculations, all of the change in pH, all the chemiosmotic hypothesis is saying it should be 150 to 200 millivolts. So where's that difference coming from? Is partly that we're using voltage sensitive dyes to accumulate, and it's very difficult to calibrate them. And it's partly that we now know the structure of the mitochondria is different. They're a closed cristae. So what you're measuring with the microelectrode is the difference between the matrix space and the cytosol. And what we really want to measure is the difference between the matrix space and the cristae space. And no one's really ever managed to get anything into that cristae space, which is capable of reporting back on the charge there. So it's definitely working at the molecular level for sure. It depends a lot on the actual structure, which only in the last few years with super resolution microscopy and cryotomography and things like that are we able to really get a handle on exactly what is the structure of mitochondria and exactly how do these spaces relate to each other and so on. And no one's ever measured it properly. So it's kind of glaring ignorance again at the heart of all of this. But until we can measure these things accurately and understand how they change over short periods of time, which may be seconds or minutes, I would think, then there's got to be a certain amount of hand waving about it. But to think that it's not the right place to look is, I think, to miss the most important things in science now. And what we're actually dealing with, I mean, there are stunning structures to the cristae membranes. We tend to just see them as a wavy line in the textbook, but they're stunningly structured. But sometimes you get amazing kind of arrays of triangular shapes and things. You see some, they're doing bioenergetics, but in what world are they doing that? There's nothing in known bioenergetics that says why they have triangular shaped cristae. We just have no idea.
So for you, metabolism is like central to bioelectricity, is it?
I think the two are connected and are integrated that way. I think they have been from the very beginning, because to my mind, for any form of selection to work, you have to have growth. You have to have cells which are duplicating themselves. And then you have some form of selection. Either they do it slowly, or they do it quickly, or they survive longer, or but you have different outcomes. As soon as you've got cells, you don't have to have genes in those cells. You just have to have cells which are capable of doubling. And if you're starting as life does on Earth with CO2, then the point of having the electrical membrane potential is in effect to get CO2 to react with hydrogen. They're not particularly reactive. They don't particularly want to react together. And cells today effectively use the charge on the membrane to force them to react. And those charges make sense at the origin of life, because what you have, as Peter Mitchell actually said in 1957, the inside and the outside of a cell, if you like, are two equivalent phases separated and linked by the membrane that's between them. And those phases are not the same in terms of their ionic composition or anything else. They cannot be. One is the living cell and the other one is the outside world. And that necessarily generates charges on those membranes, and those charges necessarily organize things. So however much it's just there. You cannot avoid it. You will have a charge on a membrane, and that charge on the membrane does something useful, even in a purely prebiotic world, which is to say it will make carbon dioxide react with hydrogen to make organic molecules inside this cell-like thing, which is now got more organic molecules inside, which would include more membrane molecules and everything else. And so it can grow. So the whole thing from the very beginning, way before we had genes, is driven by charge and structure.